Maxam-Gilbert's chemical degradation method. Before the popular Sanger sequencing came about, there were two DNA sequencing methods introduced by Alan Maxam and Walter Gilbert in 1973 and 1976. The first is known as the wandering spot analysis, which reported sequence of a whopping 24 base pairs. The second, more effective yet still limiting method using chemical sequencing. This means it used chemical processes to terminate DNA strands. These fragment DNA pieces were then run through a gel to resolve the sequence order. In this method, which is illustrated in the figure, following steps are involved. For the preparation of homogeneous DNA enzyme, restriction endonuclease is used and DNA is cut at a specific sequence. The second step, since DNA has sugar phosphate backbone, phosphate present at the 3' end of the cleaved DNA will be removed by phosphatase and replaced by radioactive phosphate using kinase. The third step, the radioactive labeled DNA fragments again treated with another restriction endonuclease and further cuts the DNA fragments. Fourth step, divide the mixture into 4 samples, each treated with a different reagent having the property of destroying either only G or only C, or A and G or T and C. The concentration of reagent is so adjusted that 50% of target base is destroyed, so that fragments of different sizes having phosphorus 32 are produced. The DNA fragments are ran through gel electrophoresis. Electrophorese each of 4 samples in 4 different lanes of gel. Fifth step, autoradiograph the gel and determine the sequence from positions of bands in 4 lanes as shown in the figure. 4 base specific chemical samples are produced. To visualize the fragments, the gel is exposed to X-ray film for autoradiography, yielding a series of dark bands, each showing the location of identical radio-labeled DNA molecules. Early strategies: shotgun sequencing and pairwise end sequencing. In the shotgun sequencing method, several copies of a DNA fragment are cut randomly into many smaller pieces. All of the segments are then sequenced using the chain sequencing method. The fragments are then analyzed with the help of a computer to see where their sequences overlap. By matching overlapping sequences at the end of each fragment, the entire DNA sequence can be reformed. A larger sequence that is assembled from overlapping shorter sequences is called a contig. From looking at the overlapping information in these 3 fragments, we can reconstruct the entire DNA sequences using shotgun sequencing. Originally, shotgun sequencing only analyzed one end of each fragment for overlaps. This was sufficient for sequencing small genomes. However, to sequence larger genomes, such as that of a human, led to the development of double-barrel shotgun sequencing, more formally known as pairwise end sequencing. In pairwise end sequencing, both ends of each fragment are analyzed for overlap. Pairwise end sequencing is therefore more cumbersome than shotgun sequencing, but it is easier to reconstruct the sequence because there is more available information. Template. Template is a very common problem area. If the chromatogram is blank, has very low signal or starts well but gradually dies out, the template should be examined. For plasmids in the 3 to 10 kilobase range, 0.2 microgram is a good amount of DNA to use. More is almost never better. It is informative to run your template on an agarose gel using some standard of non-concentration and estimate the relative fluorescent intensity following gel staining. One problem is that accurate quantification of DNA is not easy. Template composition is a factor that can cause sequencing problems. GC rich templates will generally sequence fine unless there are particular regions of very strong secondary structure. A diagnostic trace pattern for this sort of template would be a chromatogram that looks great to a certain point and then suddenly dies out. Poly A or poly T regions will often cause difficulty. The chromatogram will look fine up to the poly T or A stretch, then earlier be very noisy, peaks, under peaks or just long rolling hills of the 4 chromatogram colors. This results from polymerase slippage on the poly T or A region and is a difficult issue to resolve. Another strategy is to design anchored primers, a poly A or poly T sequence with the final 3 base being either C, G or T or G, C and A for the poly T primer. With these primers, the poly A or T stretch needs to be at least 17 bases long and special conditions for annealing, that means the 42 degrees Celsius and cycling at 52 degrees Celsius are recommended. Sometimes this works well, but it may require the user experimenting with different conditions. Finally, di and trinucleotide repeats can cause poor data. It is usually pretty obvious when such sequences are at fault.